since World War II, state-sponsored research has dominated science, as we all know. And it's with the underlying principle, the social contract, that funders, on behalf of the people, fund research with the hopes that it gets translated down to inform policy, clinical applications, and new technology. And it's a major part of scientific enterprise. And for me, as a science of science scholar, we study that enterprise mainly through what's publicly available, the scientific article, the publication, and public grants. And even that has shown us a lot about how science works, whether it's from long-term impact of science to team size, where interdisciplinary domains come from, and the career mobility of scientists. Even the scientific articles and bibliometrics have shown us a little bit about how science works. But the article is a skewed picture of the scientific enterprise. To assume that bibliometrics is what makes science science is an oversimplification of its complexity, and that we can do more to understand what goes behind science than just looking at the flawed picture of the scientific article. Because if we could look into the process, if we assume that the products of science, the article is the products of science, then we're missing the process. If we could look into the process, we could understand more about whether does the science narrate the funding or does the funding narrate the science. Or in the innovation pipeline, all the way from ideation, all the way to long-term impact, where does innovation spill over? Where does it fail? One thing about the article is that we don't ever look at failed projects because they don't make it to the article. And because I only have 15 minutes, I can't answer all these questions today, but these are questions that I am answering at the moment, some of which I will be able to give a little bit of clues about how we were able to reveal those conclusions. So I think it's very fitting that the organizers here were funded through NEPI, looking at the big science in particle physics. I am talking about the big science in biology. So I think many of you would have been alive during the Human Genome Project era, where basically tens of countries and thousands of researchers came together for the first time in biology to sequence the human genome. And it's notable for many reasons. One is that it garnered more public attention than ever before for a biology project. Before that, we were looking at Drosophila, C. elegans, in the laboratories. But second, it impacts even us today. Most of biology, modern biology research comes from most omics methods. It would not be possible without a reference genome. And that genomics as a field can be constituted down to the rise of that scientific field from the sequence of the human genome. And it has ushered in new data sharing practices that we all take for granted. And it's combined computation with biology together to make history. And so notably, as you see with Tony Blair is not mentioned here, but Tony Blair was here at this conference just through a televised Zoom. But it was mainly led through two big organizations. The Wellcome Trust at the UK, but also the National Human Genome Research Institute, which was the Human Genome Project arm of the US NIH. And you can see the leader of NIH, Francis Collins, there. And actually, that agency, we see, is one of the most innovative funding agencies inside the NIH, whether it's through the share of publications that are among the top 5% cited, whether the citation impact of them within 10 years, clinical applications from patents, or whether the research itself is disruptive. And NHGRI is consistently one of the most innovative agencies in the NIH, and so therefore in the biomedical community. But we know that it's innovative. We don't know why it's innovative. So that's why we got to look at the process. What are they doing? What are we doing that they're able to lead innovation in this way? And so we're a team, an interdisciplinary team of not just engineers, but of historians, physicists, ethicists, computer scientists, Francis Collins, the leader of the Human Genome Project here, and the director of NHGRI are all coming together to understand what it is that what led to the rise of genomics, where are the failure modes, where is the innovation spilling over, and how do scientific funding agencies work with academics, scholars, and scientists to do better science. What we looked at is that because of the notable historic nature of this genome project, they just happened to preserve many of the internal forms and documentations during this era, from the 80s and 90s and onwards. What that means is that we have the internal archive of the NHGRI contains the meeting notes of scientists that were meeting daily to coordinate a genome project. We have the handwritten notes that were taken during the correspondences of agendas and conferences that were happening. We have presentations, spreadsheets, newspapers that were marking this time. And this sits at more than 2 million pages, and it's growing by 5% every year through digitization. But as we all know, how does one person, how does a team study at scale this born digital artifact that is very difficult, as we know, to study? And so that's where we came into play. And so these internal documents are inherently different from what we know in public, that's available publicly. The RFAs and the publications that are circled right there are what we as scholars can access. They're what's available in your PubMed, they're what's available in your reporter, but their content is entirely different from what's available internally. These describe some of the many genomic projects that this funding institute has funded, and each project is of tens and hundreds of millions of dollars with thousands of researchers working together to create resources for the genomics community that led to the rise of genomics. So I can't talk to all the methodology that we use, but I'll spill a little bit over some of the things we do. One thing is that because it's a born physical archive, we have a lot of handwriting. And we have a separate ethics case study that describes why handwriting is something that's very difficult for AI to use, not in terms of technical proficiency, but also as an ethical point of what we don't know what's in the handwriting. We actually train our own handwriting model to remove handwriting, which helps with OCR for printed, but also it allows us to create a separate pipeline just for handwriting recognition. We also make good use of multimodality models that are from the research, the large research of document intelligence community that's coming in, where we can combine both the vision, the text, and the layout modalities smartly to do many different tasks, one including entity extraction, but also we can create our own synthetic documents that can help us generate synthetic training data sets that help us create new classifiers down the line. And these methods work well. Of course, because this archive is a living archive containing real people, real credit cards, real social security numbers, and some of those who are still in government and in academia today, we have to be very sure about removing and masking and disambiguating individuals from this large archive, which we do. And I'll give you one case study. We can take the printed scanned copies of emails and recreate an email network that was happening during this time. We can see that each dot here is represented by literally a paper copy of these emails, which we extract entities and link them together. And we can associate them with whether they're affiliated, which organization, which funding agency, which company during the Human Genome Project era. And we can do some really cool analysis. Let's look at some network analysis. We can look at how academia and NIH work together. This is a stochastic block model, your typical community detection method. And we can look at how people interact with each other to coordinate such a large project. So right now, what you're looking at is emails that were happening during the International HapMap project. So just a little biological disclaimer. International HapMap project was another big science, quote unquote, genomics project that happened right after the Human Genome Project. The only difference is that the Human Genome Project was a sequencing project. The HapMap project was a genetic variation project. If you're in the biology community, the HapMap project is the reason why we have GWAS, the Genetic Genome-Wide Study Association studies. But this genome project was large. It had many universities, many agencies involved. And so how does a funding agency coordinate and manage such a large project? So usually with these projects, we have something called a steering committee, which contains 5 minutes. I'm not gonna have a lot of time. A lot of people from each university. But we actually were able to synthesize for the first time a non-previously discussed group that was called the quote-unquote the kitchen cabinet. If you're from the Nixonian era, you'll know what that means. But it basically was an informal leadership circle that met right before the official steering committee to make sure that there would be no big problems that were happening in the open. And we were able to computationally, in an unsupervised way, find that in our archive. And so we can look at how does this leadership circle differ compared to the formal leadership circle? So this is a brokerage role where basically, depending on how each nodes interact with one another, we can assume a little bit about what role they're playing. You have something called a consultant where you're receiving information, you're giving it back to the same group. You have something called a gatekeeper where you receive information, but you don't give it back to that same group. But you see that, for example, the kitchen cabinet, the informal leadership circle was acting in a very consultant way that's very different from the other leadership circles that were happening during this time. I'm going to speed up a little bit because we have a lot to discuss. One of the things is that because it's a funding agency, and we can do a lot of portfolio analysis, right? And one of the things we looked at was what is a good case study for modeling decisions. So these are the 4 animals I put up to you because I don't know really the difference between these 4 animals. I know there's a gorilla in there, there's a chimpanzee in there, a spider monkey, but it's hard to know what those animals are. But these were the decisions that scientists inside the funding agency had to think about when they were deciding what money to give to which organism community to sequence their genome after the human genome. So decisions that you're thinking about in your head is the very decisions that leadership at the funding agency they have to think about how to send and share money and allocate funding. So we model that using, again, with machine learning. So we look at some of the biological features, like what's the genome size? What's the distance to the model, a known sequenced model organism? We also look at project features, like what's the team size that propose that we want this organism? Let's say you have a Drosophila community that's like, I want the fruit fly to be sequenced. Is their team size bigger than the one that proposed the zebrafish? We also look at the reputation. What are the H indices of the authors that are submitting the proposals to those funding agencies? What is the community support? Is the community large, or is it just a small, tiny community that wants their organism to be sequenced? And of course, since we're in this workshop, we look at linguistics. Like, is your proposal argumentative? Are you being repetitive with your proposals? And the main takeaway is that all these features are informative, and because they're informative, and we can model them at least in correlation association well, we can start to look at how these features affect these models. So we can look at using feature interpretability, which I'm sure there was a gentleman from the first day that talked a lot about that, so I'm glad they did a lot of the legwork for me. We can look at how these features inform the model. So I'm going to give you one example, which is that, is there a Matthew effect at play? We can see that as your maximum H index goes up, it's more likely that your funding is going to get approved. Of course, you can look at what organismal community you're coming from. Most likely, if your organism is very popular among scientists, it's going to get approved, and it confirms what we know about the Matthew effect. We're trying to maximize, funding agencies are here to maximize impact downstream, and clinical applications. And so everything I discussed is just one picture of what we can do by combining born physical archives and computational tools. So NHGRI is just one of many partners that we have in our consortium, some including federal court records from the United States or seismograms from seismology. And we basically try to translate that data using robust algorithms and cyber infrastructure to help inform policy, increase data accessibility, and answer really cool scientific questions that I'm passionate about. And so one example is that right now, today, we have shipping containers containing a lot of born physical data that could go neglected, that are vulnerable to damage. And so it underscores the importance of the work we do here, because it is important that we preserve that data for future generations of scholars and scientists. And we are looking for users and testers and collaborators with our consortium. And the one last note I'll make is that with the recent events that's happening in the NIH in the United States today, it's of note that the NHGRI was one of the proposed agencies to be dissolved in the last year. And so I just want to make a note that it would be of paramount importance to understand that this is one of the most innovative funding agencies that's ever happened in the history of science, and that it would be the data that's contained here contains a lot of scientific questions that we should answer ourselves. But thank you, and I think I'm out of time. Thank you.
Thank you, Spencer.
I'm gonna take questions one by one because I can't remember all the questions.
Sure. Let's do it like that. Are there any questions?
Okay, thank you very much. Very interesting and inspiring. So what methods did you use to increase the diversity of synthetic data that you generate? I understand you generate multimodal synthetic data for each type of your publications?
Okay, yeah, that's a good question. So the creating the synthetic data is just one part of what we did. So you can imagine like the alternative is we could take real data and hand label, right? The PDFs, the regions that we want to extract or the people, which we did. But we thought that was also very time consuming. So some of the things we did was, for example, with entity recognition, we would generate documents and put fake entities in the documents. And that you can do with an LLM. An LLM, a text decoder LLM that can generate new names that are now not real people. And we can put that in the documents and have we know exactly where it is. We know where we put the regions in the documents. Another example is with the handwriting. Though it's hard to detect where handwriting is. What we did was we took a printed document, we took real handwriting, we superimposed it. Now we have a supervised synthetic set because we know exactly where we put it, and that becomes our training set.
Okay, next was Adrian.
Yeah, thank you. And you mentioned our project NEPI, where we will try to analyze the email communication inside a large collaboration in particle physics. And if I understood correctly, you also analyze emails, but it's in the funding agency, right? The communication.
Actually, so you're partially right that it is inside a funding agency, but it's also emails that went external and internal. So we have emails that came from companies, that came from external universities, but it was preserved in the funding agency, yes.
Okay, yeah. And just what different structures would you expect if you had to guess, or if you had thought about it already?
Yeah,
From comparing a sort of a homogeneous closed group to this more heterogeneous.
Yeah, so I was I was lucky that because a single institution preserved these, actually, if I can go back, we can go to what they looked like. You can see that they follow this header, right? You have the name of the person, you have the front, the where it was sent, and actually because this is from the Microsoft Outlook, right, and the 90s Outlook, but still an Outlook, and so because it was the same institution, actually most of them follow the same format, which helps us with like the entity recognition part because it's not that structurally different. There are some emails that don't follow that, and those we do a lot poorly on, but it's the minority of them. But so we were lucky that we didn't have to answer that question of like the emails themselves weren't as heterogeneous, and that's why we actually use this as a case study because it's a good pilot set. I don't know if I answered your question. I'm sorry.
I thought about the structure, about the network that comes out when you make
the network. Okay.
So would you expect that in this kind of case with a more heterogeneous group, you expect many subgroups, many communities, and in the other you would expect a homogeneous distribution of edges and so on? Yeah,
the one thing we know I want to mention is that we saw that actually a lot of people who were interacting with the funding agency from externally had this like bridging agent where a lot of the external people would talk to the same person from the funding agency and they would spread the information to the funding agency. So we see this bridging node effect, which we wouldn't see if it was a more homogeneous group. The degree distribution didn't know anything peculiar. It follows your usual like it follows the Pareto law, right, where most of the emails come from leadership, right, from the director, the assistant director, stuff like that, which we don't actually think is reflective of actual what happened in science. This is that through the artifact of preservation, we are preserving the most important, and mostly the people who are important are those in leadership. And so we have to note that when we think about the analysis that's happening here. But that's okay, because some of the analysis we look at is just leadership, but yeah.
Okay, next question.
Yeah, I think mine is a quite general question. As I understand this workshop here, it's about also thinking about what kind of research questions we can ask to use LLMs for, and I must say I have a bit difficulties to understand your research question because so I'm from Sociology of Science, and we know for a long time, have been knowing that these decisions about, for example, research programs like NIH funding have been usually done together with scientific communities. So we have these hybrid communities between more political decision makers and or funders and researchers. And here you seem to provide data for funders only, if I understand it right, and use very simplistic indicators like H-index, where we all know that we shouldn't use such a simplistic indicator. Team size also doesn't make a lot of sense. And also, I wonder if this isn't a very specific case that you have with the Human Genome Project. I mean, nobody would say that is a particularly innovative thing. It was a lot of work, but you know, the innovation is very limited.
Yeah.
So I'm a bit puzzled about what you really want to reach here.
Okay, great questions. I'm going to try to remember everything you discussed and try to put a rebuttal for all of them. One is that one of your questions is that this is a simplistic picture. It's just containing funders, and that like we've known through sociology of science, which is great work, but through ethnographies and through interviews that and through by law that funding agencies have to cooperate with academics. I would argue that this picture is not just funders. Actually, so one of the reasons why I showed that email network is because you can see that actually majority of the nodes are not from the NIH. They are so academics from externally are working with NIH academics to create and coordinate projects, and that speaks a little bit about what these projects involve, because these aren't not just fund and grant projects; they're cooperative mechanisms, right? Legally, NIH has to provide extramural support, and it's quote and legally substantial research support. So it can't just be I give you money and that funds the project. The NIH has to step up and provide research and support structure and administration in coordination with the academics to make that project possible. And so I would argue that what we're looking at, well, it's not a perfect picture. No model is a perfect model, but some models are useful. And so this is at least the first time we're looking at. Before this, the science of science scholars are only looking at publications. They're only looking at public grant calls. Ethnographers and sociologists, they can't look at 2 million papers one by one. So we're trying to use expertise from the historians of science that are in the NIH, the archivists of historians of science, and use computational methods to scale up some of these analyses, open up that data, and help answer some of those questions. You also asked about that this doesn't have a defining question, that there is no question I'm trying to answer. And in this presentation, it might seem like that, but that's the one reason why I presented it in this way is like, think about the questions that we could answer now that this data is accessible and interoperable with machines. We all know about FAIR, right? We want to make this data findable, interoperable, accessible, and reusable. And that's exactly what we're trying to do here. So I would argue that the questions that we can answer are very important questions that we can all agree that should be answered. And that's the main point I was answering. But I will argue that one of the things that we did show is that computational methods can, without the supervision of an expert, was able to recreate some of the leadership structures that were happening during that era. So we're trying to show that computational methods have a role in not only scaling up the data, but they can also confirm or find new conclusions about analyses. There was one more that I cannot remember, but we can also talk.
Yeah, we're running out of time, so
yeah.
I'm going to take 2 more questions. Please keep it quick and short, Sophia.
Is it okay? Thank you for the presentation. I really like the idea of following the money to relate this factors, discovery, etc. My question is related to the previous question, the definition of innovation. It wasn't so clear. Do you define it based on citations or based on which features that you mentioned?
Yeah, I now remembered your question, so I'm going to answer both at the same time. So we don't try to redefine innovation as the definition. That's of debate. So what we do is we look at how other people have studied innovation, and unfortunately, because I say that bibliometrics is a skewed picture of the scientific enterprise, that papers that are cited that I showed from Nature and Science use citation impact, patent applications, and the disruption metric as your go-to innovation. I personally have qualms about using those metrics, but I just show that if I were to follow everyone else's metrics, that the NHS stands out in front of NIH. And to answer your question about innovation, I'm sorry, I lost it. We can talk after I talk.
Okay, then one last question.
Sure. Let me just say that I'm jealous that you have access to such a rich store of material. I mean, I can just imagine all those documents, but I'm wondering about the generalizability of some of your research. So what's very interesting to me is maybe some of the work you've done with the handwriting recognition, and how applicable do you think some of your synthetic data creation, your approach to it, could be for other kinds of documents. I work with 18th century manuscripts all the time, lots of handwriting. And as far as I know, no one has come up with a way of always kind of with very high accuracy identifying layouts of these documents, all the handwriting in all the different places. And then, of course, you know, the handwriting recognition as well. I mean, so in your experience with this synthetic data generation, with these much easier documents than what I work with, but still, I mean, how applicable are these some of the things you've come up with in terms of handwriting recognition?
I mean, it's true that the business documents that we look at aren't going to be translatable down line to your like 18th century, 17th century historical documents, but the process stays the same. What we do is we just take printed documents that mimic the structure that we see, and we take handwriting and we just superimpose. So for in your case, what I would do is I would take doc pages that I know to be handwriting free. I would add handwriting, and now you have a supervised set, and use that to train a segmentation classifier. And you don't need to label them because you know exactly where you put it. So that process still stays the same, and that's generalizable. And sorry, now I finally remember, this will be the last thing, is that often scholars do say that the Human Genome Project was not disruptive, it's not innovative, it built, it developed, and there was papers that talked about that. But actually, one of the reasons why I showed this last figure is that despite NHGRI, the leader of the Human Genome Project, being criticized for doing research that's not innovative, that it just develops, this metric, disruption metric, is measured by whether or not publications that come after you no longer cite the publications that come before you. And so actually, compared to larger institutes like NCI, the National Cancer Institute, or NIAID, the Infectious Disease Institute, we actually see that NHGRI is more innovative. And so actually, I showed this to add evidence to the fact that I partially rebut against the notion that the Human Genome Project and the NHGRI was, quote unquote, not innovative. And I'll leave it at that. Thank you.
Okay, thank you very much.